Hey, boys and girls. So this is not the weather report. This is something else because I'm going to take Saturdays and Sundays off from now on for my sanity. But I'm here with Otangelo, and we're going to look over a paper. And I had the right paper, right? We were going to look over that, how much of the protein sequence space, right? By Dryden, the Dryden paper. Is that correct, Otangelo? Can you hear me?
Yes.
Oh, okay. So the Dryden paper, right? I can share it.
Yes, I can hear you.
Okay. So let me see here. Application window. So I've got it highlighted so that we can go through it a little faster. And I think we can.
The Dryden paper, right? I can share it.
Yes, I can hear you.
Now we're echoing. Otangelo.
Let's see. Application window.
We're echoing. Oh, let's see. That ain't gonna work. I'm not sure. Let me see how to do this here. If I can make it a little bit bigger. Some reason it doesn't change. Okay, yeah, that's pretty good right there. I think we can see that at home. So, or people can see that. Okay. Let's see. I'm gonna turn off my cam because no telling what kind of things I'm gonna be doing with my face. Anyway, okay. So, what do you think this paper is trying to say, basically?
Well, to deny that sequence space and the odds to have functional proteins is a problem.
Okay, I'm not sure if I understood that too. So basically, okay, the abstract of the paper, or it's kind of an article. So the beginning of the paper says it is quite plausible for all of the functional protein space to have been explored by life, and that furthermore, at the molecular level, there's no role for contingency. I'm not sure what they meant by that. To be, let's see. So we've got, so two assumptions are generally made. One is that, the protein space is very big, right? So
yes.
Okay. So like for 100 proteins, we have 10 to the 130th possibilities, or for 100 amino acids.
Yes, for 100 amino acids, yes.
Right.
I think this is we can calculate this. This is uncontroversial. And that was actually my
Okay, you're roboting a little bit.
Initial reason why I linked to that paper at that opportunity when we had that conversation.
Yep. So let's see. We conclude that the second assumption is incorrect and calculate the most of the sequence space may have been explored. So firstly, okay, they say two assumptions. Okay, two assumptions are generally made. One of them, firstly, the size of the protein space. Number of possible amino acid sequences is astronomically large. And secondly, that an infinitesimally small portion has been explored during life on Earth. Okay. So those are from papers 1969, 70 up to 2005. And then they're denying that. And basically what you showed up here was the sequence space is 20 to the 100. And the way you calculate that is that there's 20 ways to pick the first amino acids and 20 ways to pick the second, and you multiply all those out and you end up with 20 to the 100, and you can approximate it by multiplying, you'll end up with 10 to the 130th. So you multiply that exponent by 1.3. Okay.
Yes.
So let's see. Okay. So it's a really big space. Okay. That's what we start out with. But then, for one thing, the problem with thinking that the whole space hasn't been explored is that the actual identity of most amino acids in a protein is irrelevant. And they figured that out because they can look like between two different humans, and they can look at our genes and they'll find that some of the space is highly conserved. It'll be identical, but then large portions of the protein will not be conserved. They basically vary within a certain range. So those things are non-conserved, and the reason they're non-conserved is because they don't make that much difference. There's let me see here if I can find the right illustration. I'm gonna stop that screen for just a second and share another one. Let's see, application window. Okay, so these are the amino acids and they have 5 different classes basically. There's also there's water loving and water hating amino acids, so it divides us into 2. Okay, but then there's like 5 divisions, so you have 5 different kinds of amino acids, and sometimes in a lot of cases it doesn't matter if you substitute an arginine for a lysine or a histidine. It doesn't make a whole lot of difference in forming the overall structure of the amino acid. The thing that'll make a difference, and I can get a drawing pad out later, is these little motifs and domains that actually do something. You know, that like a lot of the shape of the protein, like the alpha helices and the beta sheets. Now, helices are little twirly things, and they form fairly easily in amino acid proteins. And then beta sheets are like parallel; they line up in long chains, and they fold back on each other, and they form kind of a sheet. And there are several different kinds of those sheets, but basically, there's as long as you have these types substituted, it doesn't make a lot of difference for most of the function of the amino acid. So that's the point. Now, let me share that original paper again. Okay, so the actual target recognition domain. That's a let's see, prokaryotic DNA methyltransferases. They have a target recognition domain. Now, recognition, there's 2 parts usually of a protein. There's the part that recognizes or attaches to something, and then there is the little cutting tool. Like if you had in a workshop, if you had some holders and C-clamps and vices and stuff to hold something, that would be the main part of the protein. But then you'd have a little tiny tool that actually did something, you know, like something that had a metal ion in it or something like that. And that's called like the motif or domain. Those names are kind of interchangeable, as I have found, but it's basically the active part of the protein. And there's a protein called DDX11, which standing for Truth famously talks about a pseudogene. And inside of DDX11, it's a helicase. It has, I think it has 7 to 10 functional domains that each do something different. One of them is an iron sulfur domain. Another one's a dead box domain. So this one protein has like 10 different cutting tools in it. And depending on where it's used in the cell, it can do a lot of different things in different orders. So let's see. The thousands of known TRD sequences show negligible amino acid sequence conservation despite the rather limited number of nucleotide sequences that are required to recognize. Okay. So I'm not sure how to. I interpret that as meaning it has to be specific. What do you think?
Well, there may be proteins which do not require all the amino acids to be functional or to bear function, and eventually they can be substituted. But you cannot generalize that. I think there must be many proteins around which where the entire sequence probably is important in order to have functional folds.
Do you have evidence of just one? Where every single amino acid has to be exactly as it should be, or as it is?
No, not not in my mind, but I mean, the sequence is important in order for the folding to be correct. And if you don't have, I mean, about 30% of all the proteins, they require chaperones when they come out of the ribosome. When they fold, they need chaperones to fold correctly. And I mean, that is also something which has to be explained in regards of proteins which are required for life to start. And I mean, this is an additional problem, I guess.
No, they need chaperones to fold correctly all of the time. They don't need. They have a certain. There's a certain state, a low energy state for proteins, basically. I don't want to switch away from the paper again, but I can show you another little diagram here at some point. Where the heck is it? I'm trying to find it. Oh, there it is. So that would be salt bond covalent modification. Naturally, I'm going to lose things. I don't know why this thing is so screwy. Let me see here. Okay, that's hard to see. There's a table basically of things that, like, okay, an alpha helix is part of a protein folding, right? And that is a low energy state. So it falls into this low energy state. And every amino acid has a, they call it a helix propensity. So it's basically alanine is the one it likes the least or the most. So it has, basically it has a zero energy. So alanine will, if you have a lot of alanine present, you're going to end up with an alpha helix, right? And then they grade up from there. There's one at 3.16, which is proline, and that's kind of a bendy one, and that usually causes a problem. So if you get a proline, you're going to have a problem. But almost all of them are, like lysine is also a problem, and that's 1.0. But the rest of them, about 3/4 of the amino acids are below 0.5 on that, and they will pop up in alpha helices. So it's not a matter of, you won't find a paper like this, because the truth of the matter is that it's chemistry that we can tell, whether or not these things are going to fold into certain shapes or not. And these become low energy states. And chaperones or chaperonins are basically, there's a couple of them, that have developed in eukaryotic cells, and I don't think they're quite as prevalent in prokaryotes, but they just allow for a more a better statistic. You have a better, they put them in a little box, and when they're in that box, they will fold into their low energy state.
Well, there are two types of chaperones. There are ones like the Groel, and these are the barrel-shaped ones, which you just said right now, where the protein goes inside, and there it gets the right folding. But there are also the chaperones which actually accompany the proteins in order that they do not fold before the right moment arrives. So let's suppose that you have a ribosome subunit which is synthesized in the nucleus or in the nucleolus, and when it comes out of the ribosomes or to assembly factors, which then accompany the RNA strand to the compartment where it will be assembled to a higher a structure, to a more complex structure together with other RNAs. So these are also RNAs which are actually essential and necessary, because otherwise the RNA strand, the ribosomal RNA, will fold too early, and then it will be not used. It cannot be used because it has to fold at the right compartment. So these are two different kinds of chaperones which are required, and I would say that these chaperones for ribosome assembly, they are also used in bacteria and in archaea.
Okay, chaperones for ribosome assembly?
Yes.
Okay. Do you have a paper on that?
Yes, I have. I'm collecting tons of information. I can provide to
Okay,
you
chaperoning
afterwards.
ribosome assembly. So we have chaperone ribosome assembly, chaperoning ribosome assembly from Science Direct, whatever, right? Bacterial ribosomes in vitro from RNA and protein requires a heating step to rearrange conformation of the intermediate. In this issue of Molecular Cell, we demonstrated that circumvents a requirement for heating. Okay, so basically, there are other ways for these things to fold. If you have a chaperone, you're going to increase the likelihood of something folding. But we're talking about ribosomes again, and that's kind of off topic, right? Okay, how
Yeah,
modern
that was just to
ribosomes
give an
form?
example. That the only thing that they wanted to say, Speed, is that chaperones, they are not only used like the Groel, where it's like a barrel where you put it in and then it will help to fold correctly. But there are also chaperones which accompany the proteins from one compartment to the other and inhibit that the strands fold before the right moment is there.
Right, and you can also find the famous one, the creationists like is ribosomal pausing. You can find all sorts of variations in how certain proteins fold, but you're talking about one or two proteins in a, specifically, and that is kind of what we'd expect in evolution, is that you're going to end up with some situations where much more complicated or specific folding happens in one-off cases, but that's not what we're talking about in this paper. We're talking about all of the functional proteins that are possible, right? And out of those proteins, there's a lot of them that will fold all by themselves. Basically, they just naturally fold up, given the time to do so. If they're, they're in a dense eukaryotic cell with all kinds of stuff going on, there's a really, there's a chance that they'll be buffeted about and they'll end up forming globular proteins, which are kind of like big balls of mud, that sort of thing. And those get destroyed at some level. But those, the more good proteins you can have, the better. But I mean, you're talking about one or two different, like ribosomal pausing, that applies to, as far as we know, only a few proteins. We haven't found ribosomal pausing to be important overall. We like you said, there are 10 to the 130th different possible 100 amino acid proteins. How many of those require ribosomal pausing? 5? That's not a very convincing case.
Well, in regards of the ribosome, we need about 80 ribosomal RNA strands, and some are quite big, like the transferase center has about 3,000 ribosomal RNAs, which is an enormous number, and
in prokaryotes,
we have
you'll find far
always
less.
to
Okay, in
I
prokaryotes,
think this is also in
you're cutting out a little bit.
prokaryotes. It's about 3,000.
3,000 what?
3,100, yeah, about that, probably, yes. And that's an
Three-
enormous number.
3,000 what? You cut out. So I couldn't.
Nucleotides. About
Okay.
3,000 nucleotides. And that is a huge
Right.
number, of course.
Yeah, I think there's about 80 associated proteins that can form eukaryotic ribosomes, and then in bacteria you'll find 20 or 30 less. And then in simpler forms of life, basically, but none of that is necessary. So why are we talking about ribosomes? We're supposed to be reading this paper.
It was about chaperones, which I gave an example that chaperones are not used only to fold in Groel chaperones, but also they help in the transport from one compartment to the other. And yeah, but okay, back to the paper.
Okay, so Dill suggested that only two types of amino acids were needed to form protein structure, hydrophilic and hydrophobic. So that divides the amino acids into two. And furthermore, critical to find only the surface of the protein. So on the inside of the protein, it doesn't matter so much whether they're water loving or water hating, basically. So there's also this huge space on the inside of that fold where variation doesn't matter as much. So that's why they're not so highly conserved. So anyway, so what they do is they're cutting down that space in this paper. So you suggested this paper, right? What in this paper supports your case is what I'm trying to find out.
Well, since the beginning, Speed, I said that I mentioned only this paper in regards of the odds to have an amino acid chain with 100 amino acids by pure random chemical reactions, and the odds, they would be 1 to 10 to the 130th power. That was all the point that I made, which is a simple calculation. So this is not in dispute. And I mean, just that number demonstrate how unlikely that it would be that proteins would emerge prebiotically without guidance. That was the entire point. And then you guys went to look at the
rest of the paper. Yeah, the rest of the
rest of the paper where they try to diminish that. Yeah.
Okay, the rest of the paper.
Yeah,
tells
the rest
you
of the paper.
You're cutting out about every, like when you say a sentence, we usually don't get the end of it. So you may have to repeat yourself a couple times.
Okay, so basically the paper tries to diminish the number and then in the end saying that this is not a problem in regards of having proteins by natural means.
Right. That's what the paper's about. And they, and, you know, and they have an argument about inflating the size of the sequence space. So if you look at a protein sequence space, you're looking at like you're looking at 100-dimensional space, basically, which is really hard to imagine. But you can see that there are clouds in that space that are perfectly functional proteins. And in fact, if we were to compare me and you, we would find that I have a protein in one part of that cloud, and you have the same protein that works just fine for you in another part of that cloud. So we have these clouds of functionality. And then we have this, and what the point of this paper is that they're showing you, let's see, what is this one? Approximately a few thousand discrete folds that are possible. Let's see here. What do they talk about? They use a few examples where they show how basically it's not that conserved, that there's just a whole lot of different functional proteins in this space, and that with the size of the genome in the bacteria and the rate the bacteria multiplies for 4.5 billion years, basically the results are that the product of the number of cells, and they figure there are, let's see, 10 to the 30 bacteria on the Earth. Each genome's 10 to the 4, and they give you a calculation. This gives an extreme upper limit of 10 to the 43 different amino acid sequences explored. And then they explain to you why, let's say they gave a limit of 10 to the 50th protein sequences. There are a billion different bacterial species on the Earth, and they're making the case that all of these sequences have been explored in that 4.5 billion years, just with random mutation, that all of these things, all of these possible functional proteins have been explored. And then, of course, you look to genetics and you figure out with natural selection, if something popped up that was useful, it would end up in future generations, okay? So that's what the whole paper is about. And that calculation you gave up there, 10 to the 130th, that's just simple math. Everybody should know that already on this, in this group. But then they say, okay, the navigation over this space by natural selection is difficult. It could take many routes, thus result in organisms with largely different protein compositions, right? So the case they're making is the controversy with Gold, where he said that if life started over, it would have been entirely different. And they're saying that if there's any merit to our simple calculation, then protein sequence analysis provides no support for the idea of contingency, which means that gold was wrong. And if life started over again, it would end up with a lot of convergence right here. So we have convergence. Okay. So that would probably end up saying just because this space would have been folded. And then let's see, they say it's simply that this whole protein paradox are simply straw men advanced to initiate discussion in the same spirit as the Leventhal protein paradox. Hence, we hope that our calculation will also rule out any possible use of this big numbers game, which is exactly what you're doing, to provide justification for postulating divine intervention. Okay, they mentioned Dembski and Bradley. So this paper just totally criticizes your view and gives pretty good reasons for it. So,
and that's precisely where we disagree, Speed, because I think that they do not have a consistent case based on the objections that they give. Okay.
What are the objections? What are the objections here?
Well, the ones which you are outlining here, like that the sequence space can be smaller because not all amino acids are necessary. But I mean, the problem just doesn't start here. I mean, they presuppose that the 20 amino acids used in life, or let's suppose there were even less, that there were maybe 10 or so, that they were readily there and they could be shuffled in order to have functional proteins. But that isn't the case on a prebiotic Earth. And I have listed several different problems which would be prior to have even a set of amino acids that could be shuffled to begin with. And,
okay,
if you want, we can have a look on that. That is in the video which I made in response to that paper at my YouTube channel. At the end, I put there a list of different requirements which would have to be there. And in my understanding, the game is over already there. It wouldn't even get to have a set of amino acids which could be shuffled randomly in order to then somehow getting functional proteins. The problem would start much earlier.
Right. The problem much earlier is that the original amino acid repertoire consisted of only 4 or 5 amino acids like those found in the Miller-Urey experiments in the Murchison meteorite. So, and the genetic code was much simpler in prebiotic Earth. So does your case allow for very simple forms of life starting out, or are you trying to shoot for the ribosome again?
Would you like to have a look on the list of these problems? I think that would give a good
I know what your list of,
about the situation.
I'm very, very familiar with your list of problems, but we're looking at this paper, and what is your criticism of this paper?
So let's suppose that we wanted to have just an amino acid with 4 amino acids. Do you know what the odds would be to have just a functional sequence with 4 amino acids?
Yep.
What would it be?
20 to the 4th. Okay, you mean
so
functional?
What is the result?
Okay, a big number.
Now, what would be the odds? What would be the sequence space to have one functional protein just of 4 amino acids? What would be the odds? What would be the sequence space?
Okay, 4 amino acids probably wouldn't give us anything other than a little bit of RNA stabilization per Demeter. Okay, so if we're talking about, say, 20 amino, okay, there's a 15 amino acid protein that they found. So if we had 20 amino acids, okay, so we have what is it, 20 to the 20th power, right? Possible. How many of those are functional? Do you know how many of those are?
No, I am giving here the example with 4 amino acids because it is in Bruce Albert's book, and he says 4 amino acids. The odds would be there would be 160,000 different possible sequences to have just a functional. I mean, just to have an idea about the
right.
odds.
Everybody knows.
Just to have a sequence. with 4 amino acids are very rapidly going into very huge astronomical numbers. And I mean, with 4 amino acids, as you said, we wouldn't have probably anything functional. But if you calculate even a short sequence of 15 amino acids, as you suggested, it would be already in the millions.
Okay, so how many are functional?
Well, only one would be functional amongst of millions of different possible sequences.
Okay, did you read this paper? It tells you why, or do you know anything about proteins at all? Not only one would be functional out of that group, even according to Douglas Axe, a whole lot more would be functional than that. He, let's see, he came up, he had 150 amino acids, right? Okay. He said only 72, 10 to the 72 are functional. I'm picking his middle number, right? So that leaves us with 7. That leaves us with 10 to the 78th that are functional.
No.
Oh, yes. That's what his paper says. His paper says only 10 to the 72 are functional, and 10 to the 78th are non-functional. Okay, 1 and 10 to the 72 are functional, then that means 10 to the 78th out of all that group are functional proteins. Okay, you need to go read that paper again and look at it. And then look at his 1996 paper where he with the Barnase, he did a calculation and they did tests and they actually did some work with it, and they found out that 1 and 4 were functional. 25% of that space. Now you have no proof that for a 20 amino acid or 100 amino acid protein, you have no proof that just one is functional. We know of a whole bunch of 100 amino acid long proteins that are perfectly functional. And if you look at how proteins fold and how domains develop, you realize that the functional space is very large. And all we have to do is get from one functional cloud to the next. Anyway, so is there anything else in this paper?
It's about estimating the prevalence of protein sequences, Douglas Axe from 2004. And he says here in the abstract, the prevalence of low-level function in 4 such experiments indicates that roughly 1 in 10 to the 64th of power signature consistent sequences forms a working domain. And the working domain that is a size of 100, between 100 and 250 amino acids. So that is about the odds which you have. It is not 150 amino acids and then you have about 10 to the 75th power functional sequences. That's not what the paper says. It says that in a sequence space of 1 to 10 to the 64th power, just one signature consistent forms of a working domain. Just one working domain is there in that sequence space of 1 to 10 to the 64th power.
What size protein did he use? Was it 100 amino acids or 150 for his calculations?
As I said, one working domain has between 100 and 250 amino acids.
No. No, it can have as little as 5. Okay, go look at some working domains, and you'll see that some of them have like a conserved sequence. Okay, like a dead box sequence. I can show you a dead box sequence. Let me see here. Chaperoning helicase. Let me find my helicase. Wrong one. Damn. Okay, are you... So the only thing you... hang on a second. Before we leave this paper, we're supposed to be reading a paper together.
Okay, I can read here from Wikipedia, protein domain. In general, domains vary in length from between about 50 amino acids up to 250 amino acids in length.
No, but he picked a particular amino acid sequence, like it was what was it, 20 to the 100th or 20 to the 150th. I can't remember which he picked. I think it was
I would
150.
I would have to look into the paper.
Okay.
But,
I can pull up the paper. But anyway, so the only thing that supports you in this paper is this one line, and that's a simple calculation that we all knew about. The sequence space is 20 to the 100. The rest of this paper cuts that down and tells you that no, there's a lot more functionality than you know. So the rest of this paper doesn't agree with you. How do you respond to that?
well, I said already, even if you have just a protein hypothetically of 4 amino acids, then you have already a sequence space of 160,000 different possible sequences.
Right. Yeah, we know it's a big space. Yeah. We know that this number up here is really big, but that's the only number. So this is the only line of the paper that supports your case. This one little line right here.
Well, I think that my case is rock solid, Speed, because we are not talking
your
here
case is, you haven't made any case at all. You're claiming that we need. You're claiming that out of this huge space of 10 to 130, you're claiming that only one protein is functional. That's not true at all. That's totally false. If you're looking for a particular protein, then yes, then it would be very, very surprising that we ended up with that sequence. But there are probably 10 to the 65 proteins that have some function biologically, or could have some function biologically. So half of that space.
Where do you have that? Where do you have that from? Because I've not read that in Access paper. What he is saying is that one functional domain of, I don't know which sequence, which size it is specifically, which he takes in his paper. But if we are taking, like here, let's suppose that domain fold is 100 amino acids, then you have 1 to 10 to the 130th power, which is basically all the possibilities in the entire history of the universe, which is 1 to 10 to the 139th power. So this is utmost impossible. But what you have to consider, Speed, is that we are talking about just eventually the emergence of one protein, but one protein alone doesn't make a cell. I mean, we need thousands of proteins which work and interact together in order for life to start self-replicating and be alive. And if you have just one protein, you have yet nothing.
Okay. Well, let me pop something else up here. And I'm doing this primarily for the audience because I truly think that you are in a box, a thinking box, and there's nothing that's ever going to, I mean, I have no hope of getting you out of your thinking box, but boy, it would be cool if you did get out of your thinking box. Let's see, structure and mechanisms of SF1 helicases. Is that the one I want? I think this is, let me see here. Pretty sure that's the name of it. Okay, this right here is I think this is let's see, it's a particular kind of helicase, right? Okay. And these are active domains up here. Can you see my cursor a little bit? Okay. Mm-hmm. So these are active domains. One of them is this dead box right here, which are present in all helicases. So the dead box, I think, is the thing that attaches. It has, I don't know everything about these proteins, but the dead box is basically a DE, which is an aspartic acid, a glutamic acid, and then it has an X, and it usually has another aspartic acid on the end of it, but not always. Okay, they found variations. So that's 4 amino acids. Okay. And that's pretty common. I mean, if you statistically look at all of the mutations, and if you look across the whole genome, you'll find DE all over the place. and you'll find DE, X, D all over the place. It's pretty statistically easy to get there. We're looking at 1 in 20, right? Each of these is a domain. I think it has 1, 2, 3, 4, 5, 6, 7, 8 different domains. And that's typically what you find in these proteins is you'll find a lot of little domains. And these things, sometimes, some exons will be left out and you'll end up with like 4 of these little widgets. Pick and choose a few of those widgets and you got a new protein and you got a new function. And these are enzymatically functional domains, basically. Okay.
Well, the thing is, first of all, helicase, as you probably know, is required in DNA replication. It is
Not required. I don't know if it's required. How do you prove that?
Well, if you want to unwind, just a minute.
Well, some kind of helicase is, but if it just amounts to having a DEXX box or dead box, then who knows what kind of other proteins could have done the same function in very early replication. But we're talking about DNA replication, which came like probably 500 million years into the whole mix. So,
well, do you know life form which doesn't require DNA replication?
What's that?
Do you know a life form which doesn't require DNA replication?
We don't have any right now. Well, they found some traces of some life, but they weren't able to reproduce their experiments. But I mean, we don't know. Have you looked at all life? Do you know of any? Have you looked at every organism at the bottom of the ocean and determined that there's nothing simpler out there?
Well, I go with the evidence, Speed, and the evidence is that all life forms which we know of, they have DNA, they require DNA replication, and they require helicase. And if you look the papers which posit what the last universal common ancestor would look like, then you will always find that DNA replication is a part of it.
I don't buy the last universal common ancestor bullshit. So we won't talk about that. But do you have proof of what life was 4 billion years ago? If you put a particular bacteria in some sort of nutrient mix, right? And you look at it the next day, if you find out that only that bacteria is left, everything else has been killed, wiped out. This one bacteria just ate the whole, managed to multiply and take over the whole bin. If you put flu inside of you and it starts competing with your life form, it can actually eat you up and kill you because that's what life does. So just because all of the life we have now is a certain way, you don't know what life was 4 billion years ago.
Well, as I said, I work with what we know and not with what we do not know. And
what we know, right? And what we know is this chemistry that you're looking at. We're looking at these alpha helices, and I've already shown you with this propensity chart that I'm looking at, basically, that those alpha helices can form in many, like an astronomical number of those proteins in that protein space, right? So basically, this is a very complex structure. We don't need to have every single amino acid line up to get this same structure, and we don't even need this same structure to have a function. So you're just doing this numbers game, and you're doing what Nestling set up here. You're claiming that you have to have the winning lottery ticket when it turns out that 1/4 of the lottery tickets will pay out $2 or 10% of them or something like that, right? So there's a lot of you don't know how many lottery tickets are going to pay off.
Well, that is what these calculations are about, Speed. And
yes, exactly. That's what this paper is about. It's telling you that the space is much smaller than you think. That's exactly what the paper you brought shows us. So do you have another paper that counteracts this one?
Well,
other than Douglas Max, who's the creationist?
Well, Speed, even the reduction that they attempted to do comes to a number of 1 to 10 to the 23rd power, which is still the number of all the stars in the entire universe.
Yes,
mean,
and then they show you how all of those were sampled right up here. Let's see, in discussion, the navigation over this page. Let's see, result knowing on different protein conversation. Where do they claim? Where do they talk about this at the very end of the paper?
no, I think it's in the middle or so. If you put it on the screen, then
Okay,
we can read it together.
number of cells. Right. Okay, so it's right here, I think, where they talk about this gives an upper limit of conservative, and then it has been estimated as high as 10 to the 50 protein sequences out of those 100. That group had been sampled with life. So basically, that's what their calculation tells you, that many have been sampled. But they're not. This paper is not about abio. It's about Gould's hypothesis, about Gould's conjecture that life would have turned out differently if it had evolved over again.
Well, I'm opening the paper here. Okay, they say here, as an extreme method to reduce the size of sequence space, Dill suggested that only two types of amino acids were needed to form a protein structure, hydrophilic and hydrophobic, and that furthermore, it was critical to define only the surface of the protein. These two suggestions reduce the size of sequence space to 2 to 100th of power, or 2 to 33, respectively, approximately 10 to the 30th power. And that is still more than the number of stars in the universe. So, and you do not have still have a proteome, which would then somehow have to assemble together and to make metabolic pathways and so forth, and to start to reproduce and become life. So I don't see how that argument of that reduction from 1 to 10 to the 130th to 1 to 10 to the 30th would make much difference in that picture.
So it reduces the space from, reduces the sequence space from the astronomical number we had above 10 to the 130 down to 2 to the 33, right? Okay, so 2 to the 33 is what we're looking at.
Yeah, they make in between 2 to 100 power and 2 to the 33. That is approximately between 10 to the 30th power to approximately 10 to the 10th power, which is also an astronomical number. And we are still talking just about.
But look down here. Look down there. This is how many sequence spaces would have been sampled by life, 10 to the 50th, which is a much bigger number, an astronomically bigger number, far more than the number of stars in the universe.
Yeah.
Okay, that's what the paper. No, it's the opposite of what you're saying. Did you read this paper?
You said 10 to the 50th power, right?
Yep.
That's a huge number. That's more than the number of stars in the universe, Pete.
That's the number that they're telling you have existed on the Earth. That's how many came up. That's what they're telling you. That's what this paper is about. They're showing you that 10 to the 50th is bigger than 10 to the 33. You understand that, right? One is greater than the other?
They are talking about the odds. And obviously, a bigger number means that the chance to have a functional sequence is smaller, not bigger.
No, it's the opposite of that, Otangelo. They're telling you all the different possibilities. Okay, all the proteins that are sampled, not just the functional ones. And out of those, the functional ones are going to be selected for.
So how do you get this number?
The whole paper is about how they got this number. That's what the paper's about. They go through. They start out here and they show you how many different life forms were sampled down here. Okay, the product of the number of cells, and then the number of genes in each genome in bacteria to show you how many have been sampled. And like I say, this paper isn't strictly about what you're talking about, but it does show you that there's that 10 to the 50th different kinds of proteins have probably been created in life. Okay, and out of those,
first of all, as I said previously, it was not my initial intention to give the link to this paper in order to go through to the assertions of the paper, but just to demonstrate one protein with about 100 amino acids, the sequence space would be enormous. But if we go further, and if we even grant that not all the amino acids would be required to be the right ones to be functional, that eventually just a part would be required to have a functional protein, that doesn't tell you anything in regards of the odds to have life by unguided events, because there are many, many other problems that we have to consider as well. And one is that just one protein doesn't get you life. You need, as in Pelagibacter, about 1,350 proteins. But if you take the last universal common ancestor, which is proposed by Nature magazine in 2016, they talk about 500 proteins, which is still a number which, if you make the calculations, it is far, far beyond what would be reasonably possible. Even if I would give you all the amino acids, the set of the 20 amino acids in a sufficient quantity, and you could just make the shuffle, then it would be in the realm of the absolutely impossible to get a functional protein.
you're cutting out on us.
Shall I repeat what I said?
No, I pretty much know what you said. Okay, so what you said earlier was most important. You said the only thing you wanted to bring us from this paper is this one little line that says sequence space is 2 to 20 to the 100, approximately 10 to the 130 for a protein of 100 amino acids. Okay, that's all, which we knew, which everybody knows. Anybody who knows a little bit about discrete math could have done this calculation, right? And you said that the assertions of the paper, you call them assertions, even though they carefully reference and calculated things, and they have tons of papers that they refer to. And the conclusion of this paper, which you also deny, where they said that basically you are playing a big numbers game. Hence, we hope that our calculation will also rule out any possible use of this big numbers game to provide justification for postulating divine intervention. It's right there, Otangelo. So why did you want to go over this paper again when the only thing you wanted from that paper is that one cherry-pick quote, which actually is what we call a back-of-the-envelope calculation, which has no import at all? And I don't know. We're coming up on an hour, and I'm not going to get anywhere with you because you're not even reading that 10 to the 50 properly. You're reading it as the inverse of what they're saying, and I don't think we're going to communicate on this. So, but anyway,
all right.
It was fun. So what you need to do is find another paper that shows me how few functional things are in the protein space, and he pointed out a bunch of them. He actually has a bunch of them, but then there are a bunch of people that disagree with him. So like from going all the way back to, what do we go? Well, he's got papers from 2000, 2004, whatever, 1999. I think Dill disagreed with him. A lot of these early people were saying that there were, that the protein space is very sparse, and you could probably find a lot of these papers that agreed with you. 1998. So anyway, I don't think we're gonna get anywhere. So we're coming up on an hour, and I kind of wanted to close off in an hour. So if you want to go over to your channel and start something up, I'm sure that people here would be happy to come over and talk to you about it. We have Nestle.
No, Andrew Cumming invited me yesterday to talk a little bit about evolution. So I'll put here in the side chat my email. He can email me, and I'll open up a stream, and then he can and anyone else can join the stream, and we can have a talk about evolution.
Okay.
Okay.
Okay. So I think we can wrap her up.
Yeah, look, just give me a minute. I'll put here the. Okay, guys, you can send me an email, and I will return with the link to a stream, which I will start shortly in about 5 or 10 minutes at my channel. You can also join if you want. It will be about evolution speed.
Okay.
All right.
Let's see. So we're going to have a Saturday Night at the Atheist tonight at 9 o'clock, I believe. So it'd be nice if anybody wants to pop over there. And I will take a look at the chat. I have not paid attention to the chat, guys. So I apologize for that. But anyway, we're going to kill this thing and go undead. We're going undead. Goodbye, cruel world.
Okay. Bye, guys.